Today I'll be talking about solving sexual assault cases using DNA mixture evidence, and a key theme throughout will be how DNA mixtures help in solving crime. They're an aid, not an obstacle. So what is a DNA mixture? Most DNA evidence these days is a mixture of 2, 3, 5, or 7 people, all of whom have combined their DNA into one sample. You can see this on a handgun, where 5 or 6 people may have touched a gun, or on a doorknob, where there are many people, or on almost any surface. And you particularly see it in rape kits, where there's a victim, an assailant, and maybe other people in that DNA swab as well. So to analyze these sorts of mixtures, there are policies that are in place that offer various incentives. The goal of processing the rape kits, of producing DNA data from a laboratory that has the rape kits, and being able to interpret that data. But the incentives also often involve solving easier mixtures, and as a result, many mixtures are reported out as inconclusive. This is partly for how the funding for the work is done. The funding is based on the work done, not on the information produced. So there are incentives for labs to have backlogs. If you don't have a backlog, you can lose funding. And there are various biases that can creep into the process as well. So a question that comes up is, what incentives can we have for success as opposed to possibly for failure? Well, this is what DNA success looks like. The idea is to unmix the mixtures, to separate the mixed sample back into the DNA, or it's called genotypes, genetic types, of the different individuals who had contributed their DNA to the mixed samples. And there are now methods based on math and computing that will do this separation. If you have 5 people in a mixture, the computer, to the extent possible that the data permits, will separate out the 5 different DNA samples, unmix those mixtures, and afterwards comparisons can be made relative to people to determine match strength. To what extent is somebody present or not present in a DNA sample? So what are some incentives for DNA success? Well, success involves getting information from the DNA evidence. So after a crime lab has generated its data from the sample, to look at those signals and typically using computers to separate out the genetic types and produce information. Those genetic types can be uploaded to a database where they can be compared against other genotypes, say from convicted offenders, in order to find criminals. They can also be used to establish that somebody may not be associated with a crime and thereby free innocent people. If you have solid DNA evidence, that can also help win justice, both for innocent people as well as for victims of crime when someone is guilty. If you have really good DNA information, then DNA information can be used to help prevent crime. If you can identify perpetrators of crime and remove them from society, that's one way to help prevent rape, for example. So one approach to having incentives for success is to provide funding for information produced, information that's found, not just for work that's done. Maybe for other aspects like the speed, the accuracy of the timeliness, objectivity of the result. One example from the United Kingdom, where we did work over 15 years ago, is they have a policy that evidence samples must be processed and reported, possibly uploaded to databases if there's information, within a 3-day turnaround time, not 3 months or longer. So I'd like to tell you about some cases where information was found. This is a case from California. The evidence in this case was both investigative as well as used later at trial. This is the story of a serial rapist in Southern California who was victimizing immigrant women and their children, and he would stalk them, seek out their homes, and then invade their home in the middle of the night, rape the mothers, molest the children. And he was continuing with rape after rape, and until finally there was that one crime scene where he was doing what he usually did, which was tying up his victims using masking tape and zip ties. And he had taken the cell phones from all of the women, and the girls in the apartment, and one of them had hidden his cell phone, called 911, and that upset his plans, and he wasn't as careful. He ran into the road and he dropped a zip tie. And that zip tie produced a really clean DNA sample that led to a codicit that could identify him. But just having DNA on a zip tie in a road isn't a crime. But there were 36 other items that were collected across multiple crime scenes. And the results were largely inconclusive from the crime lab using the methods they had available, because with 3- and 4-person mixtures, older human review of data doesn't produce much information. The TrueAllele program was used to unmix these mixtures. Ultimately, the lab independently reproduced these results with their own system. And what you're seeing here is a table, we'll see more of these, that compares evidence items, which are the rows, a purse strap, a phone cord, a phone. The first group of 3 is from one crime scene. The next group of 3 is stain on pants, stain from pants, back of shirt, second scene. And the third scene, a bathtub handle where the woman who was raped was washed up by the rapist, as well as the zip tie that was found in the road. The columns that we see here are from different individuals. The first 2 are from one scene, the second 2 are from the second scene, and the third are from the third scene. If you take a look at a row, say the 6th evidence row, the back of the shirt, you'll see 3 numbers, 6.69, 17.42, and 8.86. This is the strength of match, measured as the number of zeros in the match statistic. So a 6 point something is a million, a 1 followed by 6 zeros. 18, for example, is a 1 followed by 18 zeros, which would be a billion. And a number like 8, shown all the way on the right, is a 1 followed by 8 zeros, or 100 million. Throughout this presentation, we'll be representing information by the number of zeros in the match statistic. But what that row also shows is that the computer was able to separate this mixture into at least 3 components and make comparisons with 2 people at the crime scene, C0460 and C0475, which represent the victim and her husband. The last column is the person who is accused with the crime, the defendant, and here we see that there are 8 matches that produce positive results from those 36 items across 3 different scenes. What's interesting, if you look all the way in the bottom right, that number 20, 100 billion, is the match statistic on the zip tie to the defendant, but that number to the left, that 4.55, is the match statistic to the victim. And that suddenly makes the zip tie probative, because you may be able to explain why you dropped a zip tie on the road, but it's much harder to explain why the victim is also on that zip tie. This is another case from New York. This was an incest rape of a man who got custody of his daughters after his wife lost custody due to drug-related problems. So he had 2 young daughters, and he had consistently raped the older daughter, I think from age 7 to age 14, weekly. And the older daughter was getting concerned that the father was becoming interested in the younger daughter. And at some point, there was an awareness that there was a problem after the children had fled, and recovered from the father's apartment where they lived was a comforter. And on that blanket, that comforter, were 5 stains. They're listed here as 5 rows. And each of those, after the computer separated out the daughter's DNA genotype from the father's, we see with these positive numbers, 4, 16, 16, 9, 16, and so on. These are relatively large match statistics, and they show that both the daughter and the father are on the same semen stains. Again, it can be hard to explain why your DNA as the father and your daughter's are on the same semen stains. So this is an example of DNA where the mixture establishes a connection between the two individuals, the victim and the assailant. Here's another case. This was a sex trafficking case. There was a woman who was abducted in Florida, brought to New Orleans, and essentially kidnapped by a prostitution ring. And during a sting operation to break up that ring, this woman was found in a very bad state, and she had a very extreme story of how she was beaten and how she was vaginally raped by a pistol. DNA evidence was collected, and crime labs don't routinely these days analyze three-person mixtures and separate them out into their different genetic types. So we did that, and here you can see what the numbers are. From the swab from the inside of the pistol barrel, there's a statistic of 6 zeros or 1,000,000 to the victim, of 3 zeros or 1,000 to one defendant, and 13 zeros over 1,000,000,000 to a second defendant. So this scientifically shows that the DNA of the victim and the two pimps were inside the barrel of the gun that she said she was raped with. And here the DNA evidence is, as a mixture, is telling the story and corroborating what the victim had told the police. This is from a recent trial I testified in Indiana. It involved a college rape where there were 3 senior students, co-eds, living in an apartment off campus. And there were some men who routinely came down to Bloomington, Indiana, where the university was, to party. And at some point, they didn't so much party as invade their apartment. They found 2 of the women, victim 1 and victim 2. And at gunpoint, they repeatedly raped them. Well, the third individual who lived in the apartment hid in a closet and called 911. While the 2 men were attacking these women, there was a third man in a getaway car. When the police arrived, the men exchanged gunfire with the police. The guns were dropped. They were eventually apprehended. And here again, the mixtures help tell the story of what happened. The defendant who I testified against at his trial is defendant B, and we see 4 numbers in that column under defendant B, 7 point something, 6, 7, 2. So these are match statistics largely in the millions, 10 millions, ones in the hundreds. And this is showing that he's associated with the first handgun, which is indicated in rows 2 and 3, and 12 in red, as well as with the second handgun, which is evidence item 13. So he's connected to the gun, but he's also connected to both victims. If you look at the second row for the handgun grip, his DNA of defendant B and victim 2 are on the gun. Same with the third row. And in fact, in the fourth row on the second handgun, he's connected with a match statistic of 7 zeros or 10 million, and the first victim is connected with a match statistic of 6 zeros. So here again, the mixtures are really good at showing that the defendant is present, it's his gun, and there's DNA from both the defendant and his victims through the mixtures that a computer was able to separate and analyze. So why might DNA prosecutions fail when there are rape kits and they're processed? There are several reasons. One is that the data may not be used. With older methods of interpretation, DNA evidence is processed and it's looked at and it's recorded, but the data is discarded if it doesn't meet criteria that help people analyze data. Mixtures may not be separated. There may be methods of analyzing the data where you don't get different genotypes that are found that can be used for comparison or uploaded to databases. If you don't have genotypes or good analysis, you're not going to have reported matches. The evidence isn't produced. And if you don't have results that could be explained, even if you have the matches and the evidence, they're not going to be well understood in court. So a key problem is the use of older methods that the FBI innovated 20 years ago, but failed to get all the information out of DNA evidence. And this has consequences not just for prosecutions, it also has consequences for innocent people. Darryl, Pinkins, and Roosevelt Glenn were exonerated in the last year or so through cybergenetics pro bono TrueAllele analysis of the DNA evidence in their case. This is a story where, going back to 1989, there were some very savage bump and rape cases on highways where 5 men were going around and attacking women. And 3 men were falsely identified through a mistake of evidence that was left at a crime scene by the actual perpetrators that had been stolen from these men on the highway. Nonetheless, 2 of them were convicted and sent to prison. Ultimately, about 10 years later, around the year 2000, DNA testing was done on some of the items that were found, but the methods were ineffective. They could identify 2 major contributors that were not these men, but what the post-conviction court really wanted was all 5 men who were the attackers accounted for, and as a result, Pinkins and Glenn stayed in jail. And 14 years later, the wrongful conviction clinic in Indiana contacted cybergenetics, and we were able to do much better DNA mixture separation, establish that there were, in fact, 5 different contributors. We used 5 methods that aren't generally used by crime labs to establish evidence-to-evidence matches, show that 3 of the perpetrators were brothers, and establish exclusionary statistics, just newer techniques, joint analysis of data. And in the end, Darryl Pinkins did walk free on April 25th, 2016, 2 years ago. Standing next to him on the left is his son, who was still in the womb when Mr. Pinkins went to jail. There's also an exoneration that resulted for his co-defendant, Roosevelt Glenn, who'd been in jail for 16 years and had been released early. And this is described in a CBS News 48 Hours episode, Guilty Until Proven Innocent. A question that comes up is that if we found 3 DNA profiles, why weren't the actual perpetrators of this gang rape actually found? And that's because the CODIS database doesn't really encourage the use of information if it's not in complete conformance with the FBI's interpretation methods and database methods, which can be antiquated and don't produce all the information. So something that we at Cybergenetics hear regularly from prosecutors, police, defenders, innocence projects, victims of crime is, why can't this information be used in databases? And I'm going to show you a little bit more why. So why does this DNA investigation fail? Rape kits are processed, but the data isn't fully used, the mixtures aren't separated, and as a result, if the genotypes that are produced aren't uploaded to databases, that is, the genetic information that could identify a perpetrator never gets onto a database. And so the database isn't searched. Even if there are 10 million potential convicted offenders on that database, if the evidence isn't getting there, then the information can't be used. So in this sense, when it comes to complex mixtures, the FBI's CODIS database is somewhat of an information failure because most of that mixture evidence, much of which does come from sexual assault kits, is simply discarded. So this is a picture describing the DNA database we currently have in the U.S. On the left side is the evidence side of the database. On the right side is the reference side of the database. If you look at the right side, all the way down to the bottom right corner, the CODIS database is really good with DNA references, with single source samples that come from one person with a very definite genotype and there's no ambiguity. And those DNA genotypes or profiles are uploaded to the database, and the alleles of those genotypes populate the CODIS database. But on the left side of this picture, the situation isn't as good. If you look at the bottom left, the methods used by many crime labs do not separate the mixtures. Well, if you don't separate the mixtures, then you're not going to be getting genotypes. And according to FBI policies, if there isn't enough information, you can't upload them to the evidence half of the database. But the methods that are being used are based on older criteria from 15 years ago that don't measure the information that's there, that modern computers can produce. So even though labs or other groups can use computers to generate tremendous information, CODIS doesn't recognize it. Even if the information is up on the database for evidence, maybe 5 to 10% of the mixtures get something up there, the matching algorithms for comparison shown by the arrow in the center overcalls a lot. It just does very simple comparisons of alleles. It doesn't work with separated genotypes. And in fact, that's the reason for the policy of the FBI to block the upload, is because their match algorithm isn't very specific. They could falsely incriminate people, and so they play it safe with their current technology by ensuring that most mixture evidence never gets onto the database in the first place. A better approach, which a TrueAllele database offers, and I'm sure other groups have this as well, it's just not used by the federal government, is to use separated genotypes. What that provides is, on the bottom left, we see that if the genotypes are separated for the DNA of each person who contributed to the mixture, then they can be uploaded to a more informative database, a genotype database. And as shown in the center, the types of match comparison algorithms that use real statistics, like likelihood ratios, can produce information about the extent of match, the sort of numbers that establish, is it 1 million? Is somebody actually present? Is it 1 in a million? They're not there. Numbers that scientifically establish the strength of association between a crime scene evidence item and a possible suspect using a DNA database. So I suppose that what we really are all looking for is a world where DNA evidence can be processed without any excuses, without any statement of, well, we don't have time, we don't have money, we don't have the database, we don't have methods, but essentially a world where all the evidence is processed and processed quickly, and that information is then available for use in criminal justice, where investigators have all the DNA evidence at their disposal, not just some of it, and they can use that in their investigations, where prosecutors have all the evidence from a crime scene collected, not just the small subset that is sometimes reported from mixtures, and where exonerations of people who are innocent also have information available so that everyone in the criminal justice system can achieve some measure of justice through science. And thank you very much.